Kia ora. I'm going to be running through a past exam question for a dihybrid cross. The great thing about these questions in NCEA exams is that they are the same pretty much every year. And so once you've done this one, you can really do any of them. So yeah, just follow along. You can find this question, 2017 question 1, on NZQA, on No Brain Too Small, or of course on our Google Classroom. This question's about pigeons. So we are looking at the likelihood that two different traits in the pigeon, coded for by genes that are found on separate chromosomes, the likelihood of those alleles being inherited together, and the likelihood of the different phenotypes that can be expressed. So the two traits we're looking at with the pigeons are the wing pattern and the leg feathering. The first thing that the question states is that both of these traits show complete dominance. In other words, one allele will completely mask the other in the phenotype. So we've got that classic dominant and recessive hierarchy. So for wing pattern, we've got two options. We've got the bar wing allele, noted for by capital B, so bar, and the barless, which is lowercase. So we can see there that the barred wing allele is dominant over the barless allele. And just like in classic monohybrid examples, you'll remember that we can make three different combinations of those two alleles. So we can get big B, big B, little b, and little b, little b. So those are our three genotypes possible. And two of those, due to this complete dominance of the bar allele over the barless, two of those will code for barred wings. And only one genotype, two recessive alleles inherited together with no presence of the capital B, will express as barless in the phenotype. Okay, so for the second trait, we've got a very similar thing. The allele for leg feathers, capital F, so fluffy legs, very cute, is dominant to the allele for no leg feathers, which is how you're used to seeing pigeons, I'm sure. No feathers. And just like before, due to that complete dominance pattern, we've got three possible genotypes. Two of them, the heterozygous individual and the homozygous dominant individual, will express the same phenotype, and that is feathers. Sorry, am I doing that right? Allele for leg feathers? Yes, it is. Okay, good. Feathers. I was just like, I've never seen a pigeon with feathered legs. Is this right? No feathers. Groovy. All right, the last thing it says is that these two genes are not linked. And just like we discussed yesterday, what that means is that the two genes are found on different pairs of chromosomes, in which case they can be inherited in different combinations depending on independent assortment during anaphase and meiosis. So let's imagine the bar allele is found on chromosome pair 1. Okay, I'm just making that up for ease. So depending on what version of that gene you've got, let's just say big B and little b, so that's as a heterozygous individual for that barred pattern. But then the gene for feathers is found on a different pair. So we'll just call this one chromosome pair 2 for the sake of it. Let's just imagine the feather allele is found down here. Okay, they're also heterozygous for this gene. So they've got a little f on one chromosome from one parent and a big F from the other. So the importance of this is that we can combine these letters in different ways depending on how the pairs line up. So being unlinked gives us the most variation and the most combinations. So that's basically what it's saying there, is that we would find these genes on different chromosomes. All right, part A. So a breeder crossed a pigeon, homozygous for the bar allele. Okay, so we know he's got a bar allele, he's got two of them, homozygous, homo same. So big B for parent 1. Okay, big B with respect to that first trait. Homozygous for the bar allele and the leg feathers allele. Okay, so he's also big F. Okay, we actually join these two genotypes together and we say that parent 1 has big B, and big F, and big F. So we're only looking at two traits. Remember, you've got two letters for 25,000 genes in your genome. So we're just looking at two genes and all the information that this bird has for those two genes. And he's actually got two copies of the same piece of information for both genes. All right, parent 2 A pigeon that has a barless wing pattern. Now there's only one genotype that allows our pigeon to be barless, so he must be little b, little b. If there was a presence of any big B's in his genotype, he would have barred wings. And the same can be said for feathers. So no feathers is given to us in the question. He is little f, little f. And if we combine those two genotypes into our dihybrid genotype, we've got, oh, what am I doing? Little b, little b, little f, little f. So this is the genotype of parent 1, and this is the genotype of parent 2, okay? The question asks us to find out the genotype of the F1 generation. So F1, the F stands for the word filial, which we have talked about, which basically means sons and daughters. Okay, so it's confusing to some people because the F1 generation is not the OG parents, it's their children. So we would call this the parental generation, and then their babies would be F1, their babies F2, their babies F3, etc. So F1 is our first set of offspring, our first set of sons and daughters. All right, let's find out when we cross these two individuals, big B, big F, with a little b, little f, what do we get? Well, the first step is to find out what kind of gametes these two individuals can make. Because remember, this is the genotype of their somatic diploid cells. So we've got to figure out what are all the combinations of those alleles that we can see being segregated in the gametes. Well, hopefully it's common sense to recognize that, well, each gamete has to possess one copy of each chromosome, right? So we get our chromosomes separating, and so our gametes are haploid. So we'll get one B and one F in each gamete. Okay, well, the only combination is big B and big F, or big B and big F, or big B and big F, etc. So this B with this F, or this B with this F, or this B with this F, or this B with this F, right? So 100% of the gametes produced by parent 1 will be holding a big B allele on one chromosome and a big F allele on the other chromosome. And then hopefully you can infer that all of the gametes for parent 2 will be little b, little f, little b, little f, etc., etc. Okay, because every time their cells divide, a little b and a little f will go that way, a little b and a little f will go that way, etc., etc. Okay, obviously I know that, oh, never mind. So yeah, we've got our two gametes there, and then we've got to find out, okay, when they fertilize, what kind of offspring are we going to get? Well, in every single fertilization event between gametes from this individual and gametes from this individual, we're going to have a big B, little b, and a big F, and a little f in every single zygote that results. So 100% of the baby pigeons made by these gametes from this individual and these gametes from this individual will be heterozygous for both traits. Notice that I put the two letters that go with the same gene together. Okay, it doesn't really matter which order you do them, and you're not technically wrong either way, but just try and be really consistent. Okay, so 100% of the offspring between this pure breeding barred feathered pigeon and this pure breeding barless featherless pigeon are going to be 100% heterozygous for both of those traits. Interestingly, because of that complete dominance inheritance pattern, 100% of the pigeons are going to have the same phenotype as parent 1 because they will be barred due to possessing this big B, and they will be feathered due to possessing this big F. So every single offspring will look like parent 1, and none of them will look like parent 2, which is interesting. However, we know the genotype says otherwise, and therefore the F2 generation could be very interesting, and that's the second part of this question. Okay, so in part A, we crossed a homozygous barred and feathered individual with a homozygous or pure breeding barless featherless individual. And we saw that the F1 generation, the first generation of offspring, would have to be 100% heterozygous for both of those traits because they can only receive a big B from this parent and they can only receive a little b from this parent. Same with F and F. The second question, or part B, asks us to cross two individuals from that second generation, from the heterozygous offspring. Okay, and like I said, NZQA repeats the same format every year with different case studies. So once we get our heads wrapped around this, it's very simple. All right, the first step to recognizing or figuring out what offspring are going to be produced by these two individuals is to think about what kind of gametes they can make. Remember, this genotype here is for the diploid cells of that individual. But when they make their gametes, there are a lot of processes in that meiotic division that will increase variation, like independent assortment, and finally segregation, which is the fact that every gamete will end up with just one option or one copy of each chromosome and therefore each gene. So all of the gametes produced by this individual will have one form of the B and one form of the F. So what are the different combinations of letters that can go together in the gametes of these offspring? Well, one kind of trick way of doing this is what I call eyebrows and smiles. So we do eyebrows connecting 1 and 3 and 2 and 4, and then we do smiles connecting the 2 middle and the 2 outside letters. So obviously you can see here we don't have any lines connecting the F's or B's to each other because we won't get any gametes receiving 2 B's and no F's or 2 F's and no B's. Okay, we get one of each because they get one of each of these numbered chromosomes. Okay, so we've got some gametes that will contain B and F, big B, big F. We'll have some gametes with big B and little f. We'll have some gametes with little b and big F. And we'll have some gametes with little b and little f. I like drawing them that order from biggest to smallest. I don't know why, but it's how I remember what to do. So that's what I did there. And obviously this is the same genotype for the second individual. So the gametes we will get will be big B and big F, big B and small f, little b, big F, and little b, little f. Okay, so these will be produced, each of them, 25% of the time. So these two would be 2 sets of chromosomes lined up, divided down the middle during meiosis. One half of that cell will become this gamete, the other half will become this gamete, and vice versa. Okay, so now we've got the gametes that this individual can make, and we've got the gametes that this individual can make, and we want to see the likelihood of them coming together and producing different phenotypes. So just like in a monohybrid, we're going to make a Punnett square, but we've got quite a few gametes to put along our axes. Okay, so I'm going to put one parent down the side and go, well, they can make big B, big F, or they can make big B, little f, or little b, big F, or little b, little f. So this is all the gametes made by one parent, and just the same, all the gametes made by the other parent, so I do it in the same order every time. And remember, this is exactly the same as if we did 2 different Punnett squares for these 2 traits. So if we did, well, parent 1 has big F, little f, and parent 2 is big F, little f for feathered, we would do a Punnett square like this. We would get one offspring, or a quarter of the offspring, homozygous dominant, 50% heterozygous, and a quarter homozygous recessive. And then we'd do the same thing for the B allele. Okay, but what we're doing here is just combining those 2 Punnett squares and looking at them together. What we see is there's so much more variation, and then if you kind of extrapolate this idea to, 25,000 genes, you can see just how many almost infinite phenotypes there are out there. Okay, so 4 by 4 this time. Okay, and you know the drill. So this square represents the fertilization event between this sperm and this egg, and the individual that results would be big B, big B, big F, just like his grandfather. Great, and then this square here represents the fertilization event that could occur if one of these gametes, remember 25% of this parent's gametes look like this, 25% of this parent's gametes look like this. This one would be big B, big F, little f. Okay, let's do another random one. Let's go here. Little b, big F, big F. Okay, you'll notice I do all the B's first and then the F's second. Again, I'm just being consistent. So I'm putting the 2 alleles for the same gene together, okay, followed by the 2 alleles for the next gene. And what we'll see is that the phenotype ratio is going to be quite interesting. We're going to get this individual is going to be barred and feathered. Same with this one, barred and feathered, because although their genotypes are different, well, they've got the same 2 alleles for the barred gene, but this individual has 2 big Fs. This one has a big F and a little f. He is heterozygous for feathers. However, that big F dominates that little f that he's carrying. Okay, so I'm going to come back, I'll finish this and come back and talk about the final bit. All right, so I've done it on another piece of paper because I screwed up. Well, it just looked really messy and I don't want to make this harder than it already is. So what you can see here is that I've completed the Punnett square for every fertilization event possible between any of these gametes with any of these gametes. Okay, so I've basically collected the letters on either side, put them together, and you'll see some of these are the same. So some of these genotypes will be the same because you'll get the opposite fertilization between the same gametes from different parents, like this one and this one, for example. All right, so then the question says, blah, blah, blah. Use the Punnett square to show the gametes of the F1 generation. So that's what you've done along the sides. So that's part of the marking, is showing the gametes. Okay, so remember these gametes come from one parent that has big B, big F, little f as their diploid genotype. And then these gametes come from the other parent, okay, with the same genotype, big B, big F, little f. And then these are the, well, diploid genotypes of all of the offspring that result. Okay, it says all of the possible genotypes of the F2 generation. Well, that's what you've done there. Describe the predicted phenotype ratios produced by this cross. So we've got to now figure out, well, what each of these individual pigeon babies would look like. So what we find is that to be barred and to have feathers, all they need in their genotype is to have at least one copy of the big B, so homozygous dominant for big B or heterozygous, so at least one of those big Bs will end up being barred, and at least one big F will end up being feathered. So let's look for all the offspring that will look like they are or express the phenotype barred and feathered. Well, this one for sure. 1, this is a big F, 2, 3, 4. I'm just looking for any genotypes with both a big B and a big F. 5. Oh, not that one because these got 2 lowercase f's. But this one. 6, 7, 8. 2 big little b's. 9. Little f's, little b's, little. 1, 2, 3, 4, 5, 6, 7, 8, 9. So I get 9 expressing the dominant phenotype. And then let's look for barred, no feathers. So at least one big B, but definitely 2 little f's because that's the only genotype to express no feathers. So big B, 2 little f's. That one, I'll do like a dotted line. And this one, if I had different colors and different papers, that would be ideal, but I don't, sorry. And this one, 1, 2, 3. So at least one big B, but definitely 2 little f's. Okay, so I got 1, 2, 3, so 3 of those. Let's look for barless and feathers. So 2 little B's have to be present to be barless, and at least one big F, so greater than or equal to 1 F. And I've got little b, big F, what should I do, square, and little b, big F, and little b, big F. Okay, 3 of those. So far 9, 3, 3. And finally, our last phenotype would be expressing both recessive traits. So pigeons that are barless and featherless. Okay, or I should have written no, whatever. Barless and featherless, well, we've got one guy. Whee! Okay, so we had to have 2 little B's to be barless, and he had to have 2 little F's to be featherless, and there was only one of those. So what we get is what we call a 9:3 to 3:1 phenotypic ratio, what these pigeons will look like. Remember, when we would do a heterozygous cross, just looking at one gene, a monohybrid heterozygous cross, okay, we would end up with 1 to 2 to 1, wouldn't we? 1 dominant to 2 heterozygous to 1 recessive. So instead of a 1 to 2 to 1, during our heterozygous dihybrid cross, we get a 9 to 3 to 3 to 1. If these traits are completely dominant, as they are in this example. Okay, so that's a kind of a comparison between the two phenotypic ratios expected when we look at a heterozygous monohybrid cross versus a heterozygous dihybrid cross. So we just need to wrap our heads around kind of memorizing 9 to 3 to 3 to 1. That's what we would expect to see in the phenotypes of the offspring, if they had enough. Okay, all right, I'm going to leave it there because the rest of the question is about linked genes and we will come back to that another time.